now we get to the general theme of what are the consequences of hypoxia. So we talked about three different ways by which hypoxia can occur. For example, we talked about ischemia causing hypoxia. We talked about hypoxemia resulting in hypoxia. And then we talked about decreased O2 carrying capacity resulting in hypoxia. So when we have hypoxia or low oxygen delivery to the tissues, what's the consequence? That's sort of the next discussion. And the key consequence is that it impairs oxidative phosphorylation. I've already told you prior, and we can repeat this here, oxygen is necessary as the final electron acceptor in the electron transport chain. And low oxygen will result in low ability to generate ATP, and low ability to generate ATP will result in cell injury because ATP is a necessary energy source for cells. So the key principle here is that hypoxia impairs oxidative phosphorylation, which decreases ATP. Now, what's the problem with having a low ATP, and why does that cause cellular injury? It's particularly when it's severe. Well, the idea here is that ATP is necessary for key cellular functions, and perhaps the most important and highest yield would be the notion that ATP is necessary for the sodium-potassium pump. Now, let's go back and remind ourselves that cells have a sodium-potassium pump, and the goal of that pump is to push sodium outside of the cell. If the pump fails, in particular here, due to a lack of ATP, then sodium will build up within the cell. And if sodium builds up within the cell, guess who's going to follow? Yes, water. Water will follow, and when water follows, the cell will actually swell. And the swelling of the cell can result in is a sign of damage to the cell. So this is one important function that gets disrupted, and I would say it's one of the very important functions, as you'll see in a moment when we discuss the next slide. The second function that gets disrupted is the calcium pump. Now, let me just tell you one general principle about calcium in the cell, and that is the goal of the cell is to maintain a super low concentration of calcium within the cytosol. The calcium in the cytosol is very, very low. The calcium can get pushed out into the blood, it can get hidden in the mitochondria, it can get hidden in the endoplasmic reticulum, but within the cytosol itself, calcium concentration is kept very low, and it's done using ATP, using an ATP-dependent pump. And so if there's low ATP, the calcium pumps can fail, and that would result in a high cytosolic calcium. And the high cytosolic calcium is dangerous because it can activate enzymes which you wouldn't necessarily want to activate within the cell. So calcium is an enzyme activator. The third disruption that occurs with low ATP is that we switch from aerobic glycolysis, which is very good at producing ATP, to anaerobic glycolysis, which then results in the byproduct of, number one, the poor production of ATP, and number two, the production of lactic acid, which lactic acid would lower the pH within the cell, and the lowering of the pH would cause precipitation of both DNA and proteins. And so this would be another problem that would arise when we have low ATP within the cell. The very initial phase of injury that occurs when there's low ATP is reversible. And so examiners like you to know what are the various findings in reversible injury, and what are the various findings if we were to go a step further and have irreversible injury. Very, very high yield, often shows up on exams. So let's take a moment to actually review this. The initial phase of injury, as I state here, is reversible. And what I'd like you to know is that if you had to summarize reversible injury in 2 words, you would summarize it as cellular swelling. This is the hallmark of reversible injury. Now, let's just understand that. Again, if the sodium-potassium pump fails, then sodium will build up within the cell. And as sodium builds up within the cell, water is going to follow, which is then going to result in swelling of the actual cell. Now, this would have consequences for various parts of the cell. For example, let's say that I have a cell that looks like this, which has these little processes coming off the surface called microvilli. Remember that microvilli are present, for example, in the small bowel, and present, for example, in the proximal renal tubule. And the purpose of these microvilli is to increase the surface area of the cell so that you could better absorb products. For example, in the proximal renal tubule, you want to absorb the things that you want to bring back into the blood from the filtrate of the glomerulus. In the small bowel, you want to be able to absorb all the food that's being digested along the brush border or the edge of the cells of the enterocytes of the small bowel. So the idea here, again, is that normally some cells in the body have microvilli. However, if these cells become hypoxic and lose their oxygen and then subsequently can't produce ATP, sodium is going to build up in these cells, and as sodium builds up, water is going to come in, and as water comes in, these microvilli are going to be the cell is going to swell up, and these microvilli are going to be effaced or lost so that the microvilli are no longer present. It's sort of like taking a hospital glove or a medical examiner's glove and blowing it up, and the fingers will disappear as you put more air into the glove. So as you put more water into the cell, these fingers on the surface of the cell are going to disappear. So that's why one of the hallmarks of reversible injury is loss of microvilli. So most textbooks will tell you, and examinations will ask you, about the loss of microvilli. Another important feature of reversible injury, it again goes back to cellular swelling, is membrane blebbing. So let's go back and remind ourselves that if this is the cell contains something called the cytoskeleton, and the cytoskeleton maintains the size and shape of the cell, but also maintains the integrity of the membrane along the surface of the cell. Now, if there is excess water coming into the cell, then little blebs can form on the surface of the membrane as they sort of pull away from the cytoskeleton, and that would be another sign of reversible injury. But again, what's the key principle? Cellular swelling. So that's the hallmark. That's why I say that if you just understand cellular swelling, everything else is sort of a consequence of that. All right, the third problem that could arise in reversible injury is swelling of the rough endoplasmic reticulum. Now, I mean, if we've got a cell, and within that cell we've sort of got this endoplasmic reticulum, and this is rough endoplasmic reticulum with ribosomes sitting along the edge, that organelle itself, the endoplasmic reticulum, is going to fill up with water, and it will swell. And when it swells, that will also be a sign of reversible injury. Now, let's go back and just remind ourselves. The ribosomes that are attached to the endoplasmic reticulum, are those attached covalently, or is it a loose association? It's a loose association. And so because it's a loose association, one of the consequences of swelling of the rough endoplasmic reticulum is that these ribosomes will actually pop off. And so when these ribosomes pop off, protein can no longer be synthesized properly, and therefore another sign of reversible injury is decrease in protein synthesis. So again, I don't think, I don't like to memorize long lists of things. I think if you understand the principle, then you can really put together the story. And that's the way I would put together the story here. The key hallmark of reversible injury is cellular swelling. That's the take home. Now, eventually, if there's damage and the damage it persists for a long period of time, if there's injury and that injury persists for a long period of time, that damage will eventually become irreversible. Now, if I would tell you that reversible injury is summarized by cellular swelling, then I would tell you that irreversible injury is summarized by something called membrane damage. That's the take-home. The hallmark of irreversible injury is membrane damage. We could take this one step further, and we will take it one step further because it does show up on exams. There are three membranes of the cell in particular that get damaged and result in the key consequences of irreversible damage. The first is very obvious, and in fact, it's so obvious that it makes for, we use it sort of every day, and you'll see that in a minute, but it makes for a nice exam question. So let's just remind ourselves that one important membrane within the cell is the plasma membrane, which is the membrane that sort of borders the entire cell. When there is irreversible damage, this plasma membrane will be damaged and will be lost. And as the plasma membrane is lost, that whatever is present within the cell, for example, let's say there's an enzyme present within the cell, this enzyme will leak out of the cell and into the blood, and then will result in that will be then a sign of the membrane damage. What I'm really trying to get at here, and the reason I'm pressing on this so hard is, think about it this way. When you have a patient that comes into the ER, let's say you're the ER physician and the patient comes into the ER, if they're complaining of chest pain and you're worried about a myocardial infarction, one of the key sort of gold standard tests, the first test would be an EKG. That would really show changes, particular changes that would be important. But another very important test would be to simply stick a needle in the patient's vein and take blood and then send it to the lab and test for cardiac enzymes. Now, when you test for cardiac enzymes like troponin, what are you really asking? What's the question that you're trying to answer by testing for those enzymes? You're asking the following. You're asking, was there irreversible damage? Because myocardial infarction means that you have irreversibly killed off the myocytes. And so you're asking the question, have the cardiac enzymes gone from the cell into the blood? And if they have gone from the cell into the blood, for example, troponin, then you would say, aha, there is myocardial infarction, and myocardial infarction is basically irreversible damage. So this becomes a very high-yield principle because you test it all the time. When you're asking the question, if you're asking the question, does the patient have hepatitis, and you're actually measuring liver enzymes, you're really asking the question, has membrane damage occurred? And i.e., if membrane damage has occurred, then there must be irreversible damage going on within the liver. So I hope that makes sense. Another problem or another consequence of loss of the plasma membrane is that the calcium, which is present outside of the cell, so much effort was made to keep calcium outside of the cell. As I mentioned earlier, the calcium now can rush in as well. So the intracellular calcium levels will increase even further. So these are the two consequences of plasma membrane damage. Another membrane that is going to be damaged is the mitochondrial membrane. Now, let's go back. So this is going to be a cell here, and we know that within the cell there is a mitochondria. We can just draw it like that. Let's just pretend that's the mitochondria. And so within the mitochondrial membrane is important because for two reasons. Let's remind ourselves about one thing. First of all, let's ask a question, and it commonly shows up on exams. Where is the electron transport chain? The electron transport chain is on the inner mitochondrial membrane. That's something to burn in your brain. Inner mitochondrial membrane. And so if there is membrane damage to the inner mitochondrial membrane, that's going to be permanent. That's irreversible. Because even if you were to give the cells oxygen, return oxygen to the cells, those cells would never be able to recover from that damage. So that's irreversible damage. It cannot be reversed, even if oxygen is returned to the tissue. So that's one consequence of damaging the mitochondrial membrane. Another high-yield consequence of damaging the mitochondrial membrane is that within the mitochondria there is a very important molecule called cytochrome C. So cytochrome C. Now, cytochrome C is present within the mitochondria and is actually kept very, it's carefully guarded. Why don't we put it that way? If cytochrome C leaks out of the mitochondria and comes into the cytoplasm, it has the ability to activate apoptosis. And apoptosis is a mechanism of cell death. So another consequence of damaging of the mitochondrial membrane is that cytochrome C can now leak out from the mitochondria into the cytosol. And when it leaks out into the cytosol, it will activate apoptosis, causing cell death. So that's another consequence of mitochondrial damage. A third type of membrane that is damaged is the lysosome. Now let's go back and remind ourselves that if this is a cell, one of the organelles within the cell is a lysosome. The lysosome is sort of the recycling factory of the cell. It contains lytic enzymes that have the ability to trash molecules into their basic building blocks. Now imagine if the enzymes present within the lysosome actually leak out into the cytosol, those enzymes are going to start trashing the cell itself. It's going to basically digest the cell. So that would be one problem. And that's only further added to by the fact that that calcium has already come into the cell, and calcium is an enzyme activator. So the presence of calcium and lysosomal enzymes within the cytosol becomes a very, very dangerous circumstance, which obviously would be another consequence of irreversible damage. So again, these are the high yields, and I don't memorize these things. I'm really actually just sitting here talking off the top of my head. I don't have any notes in front of me, and the reason I can talk off the top of my head is because it makes sense. If you just remember membrane damage being the source, being the key hallmark of irreversible injury. Okay, the end result of irreversible injury is cell death.